Last class, what you are completed? That is Bryophyta in Markhamshire you are completed.
Yes, sir.
Yes, so now we are going to discuss about in Anthoceras. Okay, so you see here the first one that is in Markhamshire, Pellia and Markhamshire we are last class completed. So now we are going to discuss about in the second topic that is in Anthoceras. See here, this is Anthoceras classification first. The division it is in Bryophyta, class it is in Anthoceratopsida, and third one that is in Anthocerotalis, family that is of Anthocerotaceae, genus that is in Anthoceras. So you see generally the Bryophyta three classes is there. Okay, one it is in the classification of in just 1 minute. Yes, so Hepaticopsida, Anthoceratopsida, and Bryopsida. Total this of three classes entire the bryophytic groups. Hepaticopsida. Already last class I completed what is Hepaticopsida. So now I'm not going to detail about in that one. So now we are going to the second topic that is in Anthoceratopsida. You see first what is the differentiated between an Anthoceratopsida and Hepaticopsida? So Hepaticopsida called that is in liverworts. So Anthoceratopsida here we are saying that is in hornworts. So what is the meaning of horns? So when we are looking the plant, see looking like this one. Okay, here this one is a gametophyte. These are all things this are in sporophytic stage. The sporophytic comes to outside horns like structures. That's why. So we are saying that is in hornworts. Hornwort that is in Anthoceratopsida. Okay. So what is the general characters of an Anthoceratopsida? How can we identify in Anthoceratopsida? Okay. Yes. So you see here, almost all this Anthoceratopsida is a cosmopolitan distribution all over the worldwide. We are looking in Anthoceras. So commonly these Anthoceras is a tropical plant, a tropical and a temperate regions of the world. Near about this Anthoceras, which is of 200 species is there. In our India, just only 25 species are reported. Okay, so in the 25 commonly three species is there. That is of where it is in Himalayas. Okay, Anthoceras Himalayas, Anthoceras of Erectus, Anthoceras of Chambensis. These are all three. Is mainly we are looking in our country. Yeah. So here the Anthoceras, it is a small group of bryophytic group. In spite of it is advanced characters is there. Okay. It is a very small, only one family is there in Anthoceratopsida. But in spite of, so here some of the advanced characters is there. So what is the advanced characters means? The sporophyte which differ from the liverworts. Here the sporophytes comes to outside the gametophytic area. Okay, that's why the popularly known as we call that is in hornworts. We are saying that is in. Okay, yes. So what is the thallus structure of in Anthoceras? Here now you see. So this is in thallus structures. The thallus it is a same almost all whatever you are looking, whatever you are observing in Markhamshire, same type of the structures you are looking in the thallus. The thallus, but Markhamshire we are looking at midrib. Here no the midrib. Polygonal shape is there. Dichotomous branches is there. Okay, but you see here the thallus. When you're looking at the thallus, there is no which one that is of airports. And also one is the most important. When you're looking in Anthoceras thallus, in between cavity structures is there, cavity structures. In that cavity structures are contains what type of, that is of blue-green algae is there, blue-green algae. Okay, some type of Phonastoca, Renabina, the type of the algae we are looking in where that cavities. The sex organs is a common point, Archegonia and Antheridia and Archegonia. So this is a little general characters of an Anthoceras. And now we are doing the detail about of an Anthoceras. So here now you see this of an Anthoceras thallus. Okay. So the thallus, it is a dark brown, small prostrate. Okay. The thallus, when you're looking in the thallus, this thallus, it is a total little bit dark brown color is there. Dorsiventrally side is there, dorsiventrally structures is there, and it is a prostate system is there. And next now we are lobule-like structures is there, lobule-like. Okay. So you see here when you're looking in the thallus, here when you're looking in the thallus, one lobe-like structures is there. Dorsal surface is very smooth. Okay. And there is a midrib of absent. The contents of elongated sporophytes, including one invalid care is there. Means the gametophyte is looking like invalid care at the base, vertical surface of bears only, smooth walled rhizoids is here, rhizoids. Okay, that is are absent. And Markhamshire, we are looking the trabeculate rhizoids is there, but here it is in absent. So this is the thallus structure of Anthoceras. Yes. So next now we are going to once again the thallus structure, dorsiventrally thallus. Okay, lobule-like structures is there. Dorsal side and ventral side. Dorsal side is a very smooth. Midrib is absent. There is no air pores. Okay, trabecular rhizoids are absent. The smooth rhizoids are present in this one. Yes. So next now you see here internal structure of the thallus. Yes, this is the internal structures. You see, variation is most important. When you're studying in Markhamshire thallus structures, so already you know the Markhamshire thallus is a three regions is there: the epidermis, photosynthetic zones, and which one is of storage tissues is there. So particularly when you're looking in the Anthoceras, so here we are looking in only two structures is there, two structures. One it is in epidermis. You see number one, this one is epidermis, and here now you see this one, this is in lower epidermis. Okay, upper epidermis, and second that is of in lower epidermis. The upper epidermis, here it is in a single layer. There is no any intracellular spaces is there. Okay, so in between there is no any air pores are there, no other structures. So simple structures, single layer. Okay, so you see here. And next one, this of in lower epidermis, here it is. Okay, so these lower epidermis, it is also here. Taking one special type of the structures is there. So this of in smooth called rhizoids is there. Smooth type of rhizoids is there. And next, below the upper epidermis, above the lower epidermis, all together. Okay, so which is of in this of in total parenchyma tissues is this. Okay, all this one is total, it is of in parenchyma tissues is this. Okay, smooth and here in between of these parenchyma structures. So here we are looking one special type of cavity structures is this. Okay, we call it the mucilage cavities. Here these mucilage cavities are contains different type of blue-green algae, Phonastoca and Anabinas. So here this is the internal structure of an Anthoceras. So once again, see here a very simple structure of an Anthoceras. Yeah, Anthoceras structure, it is only two parts is there. One it is in epidermis, second that is of in parenchyma, parenchyma cells is there. The epidermis, it is a two types is there, upper epidermis and lower epidermis. The upper epidermis is a single layer, There is no any intracellular spaces is there. Lower epidermis, so here also there is no any spaces, any space is there. And this here from here to we are looking one smooth colored rhizoids is this. And in between of lower epidermis and upper epidermis, you are looking total this of parenchymatous. So these parenchyma means each cell is contains, this is a special characters. No other bryophytic characters. You are not looking this type of the structures. Each cell is contains one proteinaceous structures. So these proteinaceous structures that is of pyrenoids. Okay, this is of pyrenoid. This is like this is of an algae type of characters. Each cell is contains pyrenoids. Okay, so proteinaceous structures, and in between of these structure, so here we are looking mucilage cavities is this. So here these mucilage cavities are contains blue-green algae. Okay, nostoc and anabina. So this is the internal structure of an Anthoceras. Yeah. Next, now you see here. So we are going to the reproduction systems. So generally, the Anthoceras, the reproduction system are three types is there. So one it is in vegetative, and second that is of in generally asexual is absent, and third second that is of in sexual reproduction systems. So vegetative already, so you know that one. Vegetative reproduction takes place by the mean gemma. Okay, so means tubers persistent growing the apex is there. Some gemmas, one special type of the structures is there, germinated. Okay, so when break it the parent body, automatically that is produced a new plant. That is a vegetative reproduction system. You see now here, these are all the vegetative reproduction system. Tubers like structures is there, gemmas like structures is there. Okay, so this is after the asexual reproduction system or vegetative reproduction systems. Yeah. So next now we see a sexual reproduction. This is the most important. Here now this one. Sexual reproduction systems. Okay. Here, see when you're studying in Marchamtia, the reproduction system of Marchamtia means the antheridia comes to outside the gametophytic stage. Gametophytic comes to outside the gametophytic structures. But here, the reproductive organs, reproductive parts are embedded in the thallus. That is the most important here. See here, this is totally thallus. This is totally thallus. So these structures, so these structures we are saying that is in here this one antheridias, group of antheridias. Marchamtia only single antheridias is there, but here the group of antheridias are embedded in the thallus, Okay, so this is of in Marchamtia, this is of in antheridia. Okay, antheridia develops of endogenously inside the antheridial chambers. An antheridial chamber many contain single antheridia, and here yes. So how the development of an antheridia, that is most important here. So whatever see the previous we are completed. Now for example, this is the thallus. This is the thallus. In this thallus, so first, okay, so where the first one here it is in one cell, first one cells. Okay, so we call here it is in superficial thallus cells, superficial thallus. One cell, for example, this one is Anthoceras thallus. Here, one of the sections, one of the part, one of the cell is going to divide it. Okay. So where it is in, see here, the antheridia developed of endogenously inside the antheridial chamber. An antheridial chamber contains very single or group of antheridia. Okay. And next now you see here. Yes, so first from here, This is an antheridial initial cell. We are saying this of antheridial initial cell. So here these antheridial initial cell is divided first mainly two divisions, upper part and lower part. The lower part always it is attached to where the thallus. Upper part it is going to metatic divisions. Okay, so 1 to 2 cells are divided, 2 to 4 cells, 4 to 6 cells are divided. So when this metatic division is completed, one structure we are looking in this area. Okay, so above the structures, one epidermis structures is there. We call that is in jacket layers. We call that is in jacket layer. So here now you see this one. Here it is in jacket layer structures is there. So after this one is we call it stalk-like structures is this. And below this jacket layer, here we are looking. Okay, that is of in reproductive cells. We call that is in androgonial cells. Androgonial cells. Okay, so here the first one cell, that is a primary androgonial cells is there. After the primary androgonial cells, after the development, so these jacket layer are destroyed. The spores are comes to outside in antheridial chambers. Okay, so this is an antheridial development. Similarly, second one is archegonia. So almost all similar structures in archegonia. Archegonia, it is a flask-shaped structures is there. Flask-shaped structures. So here this one it is in also embedded in the thallus. This one here it is in also embedded in the thallus. The thallus it is in embedded in the which one, that is of in archegonia. Same archegonia. Archegonia develops in acropetal fashion from superficial dorsal cell laying close or growing apex. Here also same. First now one cell is there here. Sir, these cells are divided to equal parts. Okay, one it is in a base, above in apical cells. So these apical cell is going to modified as a archegonia. Archegonia shapes is this. Now you see here, this one, this is in venter canal cells, and here it is in neck canal cells is there, and next one this is of in cover cells is there, cover cells. All this structure is embedded in the thallus. Okay, so in between of venter canal cells, the formation one special type of the structures we call that is of egg cells, female reproductive organs. Okay, so same base and upper cells. So upper cell, how the metatic division is going on, that much of development we are looking. Venter canal cells is, and neck canal cells is there, and cover cells is there. After the development, it is looking like a flask-shaped structure. Okay. Yes. After the development, sir, these female reproductive organs is produced to some type of the chemicals. So these are all the chemicals are coming about the cover cells. So you see, the spermatozoids are comes to outside the antheridial chambers. It goes to by the water, by the wind, by the whatever it may be that is in. So when goes to like this, here the female reproductive organ, some chemicals are produced is there. So attract of in this with the male reproductive organ, it goes to where the female reproductive organs. Okay, so many male reproductive organs are going where the female reproductive organs, but all the female so all the male gametes are not fertile. Only the female is taking only one male reproductive organs. So now you see we call this is of fertilizations. So when entered where the female reproductive organs, the formation that is in fertilizations. So after the fertilizations, so here formations zygote. Here formation in the zygote. So now the one of the life cycle is completed. You see here, this is in a gametophyte. So this gametophyte has terminated male reproductive organ, and second is of female reproductive organs. The male reproductive organ is produced through the male gametophyte, that is of spermatozoids, anthrozoids. So these spermatozoids, anthrozoids goes to where the female reproductive organs. Okay, completed the fertilizations, formation in the zygote. So these zygote is produced to the next generations. Next generations. What is that one? Sporophytic generations. So now here this is the most important sporophytic generation. You see now here this one here it is in a sporophytic plant body. So generally we know very well sporophyte is a three stages is there: foot, seta, and the third one that is of capsule. Three types of stages is there. So first one, foot. The foot it is attached to where the thallus. Second one, in anthoceras. So before going that one, you see here, this is a fin. Okay. The mature sporophyte as base spot, it is a foot. Yeah. After the foot, so the last class, so we are completed, that is a seta. Instead of seta, so which one that is in here we are looking, that is in special, one of the special type of the structures is there, that is in meristematic zones. So what is the meaning of meristematic zones? Meristems means always it is produced to the new plant, sorry, new cells produce the new things that is that. That's why we call that is in meristematic zones. We are saying here it is. Okay. Yeah, third one that is of in capsule. Third one that is in capsules. But how the development of in this sporophytic generation means here, you see, the first, okay, when formation the zygote, okay. So this zygote is a main, which there is a first part of the sporophytic generations. The base of developing sporophyte remains protected by the fleshy sheath formed by an, that is an upward growth of completely sunken archegonial tissues in this. Okay, archegonial tissues. Yeah. Next point here. This one first it is in a base. Zygote. Zygote is divided going to the divisions. So when it is divided, first one we call that is in a foot. Okay. So here we are looking some type of the divisions is there, metatic divisions is there. So these metatic divisions, second system is going to modify as a meristematic zones. Okay. So these meristematic zones, always it is produced to the new type of tissues, means, so when it is produced to the new type of tissues, it is what happened, some of the photosynthetic pigments is there in that one. Here these plants are prepared to the own food. That's why we are saying this is in second step, that is of meristematic zones. And next one, third one, that is capsule. Capsule means here it is, see this one here it is in a, we are saying there is of capsules. From here this one is a foot is there, foot. This one here it is in a meristematic zones, and here this one is of capsules. Okay. So here, so first type of cap is undeveloped of spores is there. So this capsule is contain the spores. From when you are coming from here to here, a little bit developed structures. The spores are a little bit developed is there. So from here, this often mature spores is there. How take the food material from the thallus, that much of development they are looking in this area. Okay, this is the first premature spores is there. Okay, here it is in going to development spores is there. This one total developed spores is there. Okay, after the development, so these spores are comes to outside the these sporophytic areas. So all together we call this the sporophytic stage. Okay. Yes, so once again, Anthocera sporophyte. Three stages is there. One, it is in a fruit. Second is of meristematic zones, and third one that is in capsule is this. Food, it is attached to with the gametophytic stage. It is taking from the food material from the thallus, and when it is taking the food material, it gives to where the second step, that is of in meristematic zones. Okay, so these meristematic zones is always it is produced by new cells, new cells. That's why here going on to the how much of cells is produced, that much of development we are looking in that area, because it's meristematic zones. So here, and third one, this is in capsule. Third one is in capsule. In between of capsule, we are looking at the columella stage is there, and some of the here you are looking which one that is of in elaters is there. Elaters is also most important. Okay, so this elaters, it is forced to with the and it is of spores. So these spores are comes to outside in this area. Yeah, these are sporophytic generations. Okay. So you see here, this is the development of in spores development. When it comes to outside the sporangium spores, this is the development. Okay. So here A, this one is a spore is there. So it is taking from the food material where the air, that is the development we are looking in this one. Okay. So A, B, C, D, E, F, G, H, and here it is in total formation of the gametophytic stage. Okay, gametophytic stage. So this is the Anthoceras thallus. Here it is in Anthoceras thallus. Okay. Yes. So next, now you see life cycle of Anthoceras. Here this one, this is of in thallus in Anthoceras. Stuff in gametophyte adult thallus gametophytic stage is there. The gametophyte, here it is in, see, first one is of in male reproductive organ, that's Antheridia. Antheridia produce anthrozoids. Similarly, female Archegonia, here it is in egg cells is the egg. After the fertilization forms a zygote. The zygote is going to modified as an embryo. Embryo, it is not true embryo, but like embryo. So this embryo is produced to the sporophytic stage. Okay, sporophytic stage. And spore mother cells. Okay, here this are in spores, germinations. This are in Anthoceras thallus. So here, this is the life cycle of in Anthoceras. Okay, yes. So next, now you see here, this Anthoceras. It is the most important topic in entire the Bryophytic, smallest, and some of the advanced characters also are looking in this one. So you see here, the Anthoceras is a cosmopolitan species commonly known as horn liverworts. Okay, so it is growing only with the moist and shady places. The plant body, it is a dorsiventrally fleshy thallus without prominent midrib is there. Only smooth-walled rhizoids are present without scales, and rather than that is of trabeculate rhizoids. The gametophyte bear the sex organs, that is Anthoceras may be homothallic, and second that is of heterothallic. The anther and archegonia is embedded in the thallus. Dorsal region is there. Antheridia occur single, sometimes single, or there is often cluster-like structures is there. Okay, so where the closed of cavity is called of antheridial chambers is there. Antheridial chambers. So the archegonia produce of biflagellate spomes. The antheridia produce biflagellate spomes is there. Archegonia are also embedded in the thallus. Okay, archegonia as a roostet of cover cells, and 4 to 6 neck canal cells, ventral canal cells, and in vitro formation to the egg. So maturity, these cells accept the egg degenerate and give me to the sperms to reach where the egg. So this is a little again structures of an anthoceras. So after the fertilizations, so fertilization takes place by the sperms and egg, and diploid zygote is formed. The zygote by cleavage develops into embryo. Embryo develops into the sporangiums, means sporophytic stage, which differentiated into fruit, okay, and meristematic zones, and third one is of capsules. The fruit is a specialized in hastudium-like structures. Is a absorb minerals, food minerals from the gametophytic plant. The capsule is a with layer of jacket cell, orchisporium, as differentiated in the spore mother cells by that is of in meiosis produced the spores. The spores are liberated out when the capsule and since the spore develops into young gametophyte of anthoceras. So this is the once again small introduction, a small summary of anthoceras. Yeah. Next, now you see we are going to one more important, that is of third one. Just one minute here. Yes. Now we are going to yes. So now we are going to so next now we are going to discuss about in the third one, that is in Today, next now we are going to one more important topic, that is in third one, where it is in Bryophyta. So Bryophyta, it is a advanced group of in entire the Bryophytic groups. So in our topic, that is in Spagnalis. That one is here, it is in Spagnalis. Okay. Yes. So when you are looking in Spagnalis, just one minute. The Bryophyta is advanced group of in entire the. Hello, please respond, Amma.
Yes, sir. Yes, sir.
It is visible.
No, sir.
Spagnalis. Now, next we are going to Bryophyta. Bryophyta, it is a largest group of in Bryophyta. So near about it is consists of 660 genera is there, and 14,500, near about 14,500 species is there in all over the world. So this Bryophyta is also cosmopolitan distribution is there. Here I'm sharing where it is in just 1 minute. Yes, this is the plant of sphagnum. Is it visible?
No, sir.
Not visible.
No, sir.
Okay, can I explain? See here, this is of in sphagnum plant. So the sphagnum, it is a advanced group, and you see now last class were completed Markamsha and Anthoceras. So Markamsha, it is a thallus-like structure is there, and Anthoceras also thallus-like structure is there. So when you are looking in sphagnum, it is a little bit plant-like structure is this. Not true plant, but here it is in a plant-like structures. Okay. So this is classes. This is a classification of in sphagnum, Bryopsida, so subclasses of in Sphagnadium, Spagnalis, and Spagnaceae genus, that is often sphagnum plant. The sphagnum, we are saying here it is moss plant. We are saying this is often moss plant. So here we are looking from here to here. This is in gametophytic stage is there. Okay. So here now you see this one. This often sporangium, sporophytic stage is there. Sporophytic stage is there. So what is the plant structure of in external structure of in sphagnum? So the young gametophore here, this is the gametophytic stage. From here it is in sporophytic stage. Both together we call that is in gametophory stage, gametophores. Okay. So here this one, this is in gametophore. So this gametophore is contained from here. From here it is in rhizoids is there, root-like structures is there, root-like structures. Okay. Yes. So next now you see here, this one from here it is in stem-like structure is there. So this stem-like structure is contained. Stem-like structure is contained, and which one that is often leaf-like structures. Okay. Here we are looking at often leaf. Two leaf is there. Anthoceras and Markham, there is no leaf-like structure. That's why this is in advanced group of in bryophytic plant. Bryophytic plant here, this is an advanced group. Okay. From here we are looking in tip of the branches. Tip of the branches here we are looking, that is often reproductive systems. That is often sporangiums. We call that is often sporangiums. Antheridial chambers is there, that is often antheridial. So here now you see this often external structures is there. Okay. Here the mature gametophore consists upright of perennial stem bearing the leaves and leafy branches, may grow up to 2 meters or more than 2 or 3 meters is there. So here the leaves are small or arranged 3 vertical rows. For example, this is the stem. So here we are looking, okay, vertical rows, young condition, but the maturity of the stem converted in 2 rows. The leaves are sessile, apex acute. Okay, so that is often without midrib. The leaves is with a single layer of cells. Each cell of young leaf divided in asymmetrically and forms a small cells and large cells. Okay, the large cell is divided to form a large and a small cells. The 2 small cells are narrow, while the third large cell is grow in directions and becomes highlands. So this is the external structures of in sphagnums. Okay, and next now we see here, when you're looking at the internal structure of a sphagnum, the internal stem structures means when you're taking the stem section cutting of in sphagnum. So generally here it is in 3 regions is there. So where it is in here actually, there is no diagram in spite of. So when you are taking the section cutting of sphagnum stem, it is a 3 regions is there. One it is in epidermis. Okay. So second one that is of in total parenchyma, and third generations here one special structure is there, that is in central cylinders. Okay, the central cylinder, that is a third position of in central part. Okay, the first one that is of in epidermis. So outer with the small, narrow, thick, elongated parenchyma cells and the outermost layer of cortex. So which is made up of large cells. Some species it is in 2 or 3 layers is there. So here this is internal structures of in sphagnums. Okay. So center part of the cell we call the central cylinder. So these contain special type of the tissues is there. So it is taking food material from the soils. Yeah, next one here it is in reproduction system of sphagnum. Sphagnum takes place by vegetative and sexual methods is there. So vegetative stage, so we call that is in where it is in Protonema. You see now here formation, which there is often boards is there. Sometimes you are looking the boards like structures is there. When the older leaves, vegetative reproduction also takes place, which one that is often where the gemmas like structures is there. Okay, so gemmas are produced in small type of vegetative structure that is often protonema. Juvenile stage is there, protonema stage is there. So these are all things of in sex vegetative reproduction systems is there. So the sphagnum vegetative reproduction systems, it is produced to the birds is there, and also okay, so gemmas is there, and also you see these gemmas are germinated on special type of the structures we call that is often protonema stage is there. Yes, so next now you see here the reproduction system of in sphagnum. Here the reproduction system of sphagnum, so already you know very well that is how it is a monoecious or dioecious plant. Antheridia and Archegonia are born different branches or same plant. Different plant or same branches. Okay, Antheridia and Archegonia. So, for example, yes. For example, in this plant, so this plant, tip of the branches, so one of the section is produced to the male reproductive organs, and other one is female reproductive organ. Sometimes single plants are produced with two type of the plant. That's why these plant, a sphagnum plant, it is a dioecious is there and monoecious is there. Okay, all the Antheridia were coming to tip of the branches. That is important point here. Okay, na? Yes. So now you see here how the development of an Antheridia. See, almost all bryophytic structures are similar. The first now you see here this in this area. Here we are looking at super initial cell is there, super initial cells. So here that super initial cell is going to which one is of metatic divisions. For example, now you see here this one. Okay, we call this of superficial antheridial initial cells. So these cells are first going to divide it. Okay, transfer divisions, small filamentous are formed. So these cells first it is divided into two parts. Basal part is attached to where the gametophyte stage above 40. So going to development I say so antheridial chamber. You see this is the development of an antheridial chambers. Okay, antheridia develops from the single superficials antheridial initial cells which undergoes several transfer divisions and small filamentous is formed. The terminal cells are filament functions as an apical cell which divides asymmetrically into jocket initial and larger sister cells. By further divisions, the larger cell becomes the primary androgonial cells and jocket initial cell with long stalk is this. Okay, here. Say this is the development here. It is divided in three after 1, 2, 3, 4, and here of the which one is of male reproductive organs. Okay, here this one in Antheridia. Yeah, this much of development you are looking. You see now here it is of in when the axis like structure, the X structure is there. Here it is in androgonial cells is there, androgonial cells, jocket layer is there. And next now you see this one, this is of an androgon. After the development, we are looking like this. are looking like this. Okay, it is divided into, after the development, the spores are comes to where it is outside the antheridial chambers, outside antheridial chambers. So when it comes to antheridial chambers, you see we are looking like this. Blepharitis, flagella-like structures is this, spermatozoids. So these spermatozoids are goes to where the female reproductive organs, where the female reproductive organs is this. So this is the development of in antheridia, and here now you see this one. This is of an archegonia development. Same here also. Super initial cell is there. It is going to divisions of this one. It is divided from the filament of 4 cells. The terminal cells of the like the filamentous and becomes enlarged by 3 vertical 1, 2, 3 vertical rows, forming 3 peripheral jacket initial structures is there surrounding to primary axial cells. These cells divided transferally to outer cover cells, and which one that is of inner central cells are formed. So after the development, here also same. Now you see here this is of in female reproductive organ, that is in archegonias. Okay. Yes. So when the spermatozoids comes to where the female reproductive organ entered inside the female reproductive organ, formation is the fertilizations. Okay. So after the fertilizations, the formation, this is of in zygote. Okay, this zygote is germinated to the new type of new generations, that is of sporophytic generations. Here now you see this one. Archegonias. So here now this one is sporophytic generations. So after the fertilization, formation the zygote. So this zygote is produced to the new plant, sporophytic stage. So here this is of in sphagnum. Sphagnum cycle. So next life cycle of the sphagnum is also very simple of sphagnum. So here this is in gametophyte. The female branch is there and male branch is there. The female branch are Archegonia, Antheridia, egg, fertilizations, forms a zygote. Zygote is produced to the new sporangiums. This sporangium is produced to the new sporophytic stage. This is the life cycle of in gametophytic stage. Okay. So this is of in sporophyte. You see once again the sphagnum. The sphagnum, the young gametophore of sphagnum with upright stem with 2 rows of leaves grow to up to 2 meters, or sometimes we are looking for 3 meters also. The branches of limited growth at apex and elongated branches about 3 to 8 occur on the stem. The sphagnum may be sometimes monoecious or that is in monoecious, that is of dioecious, and antherial branches appear at the apex. Solitary antheridia is spawned in the axis of each leaf. Antheridia is a globo structure with a jacket layer and inner androgonial cells, which develops into where it is in bioflagellate antherozoids. So similarly, Archegonia are born on the short branches. Archegonia consist of many, which one that is in where the neck canal cells and venter canal cells and an egg. During the fertilizations, antherozoids unite the egg and forms the zygote. After the fertilizations, the zygote develops into upper cell and Another one that is in lower cells. Those lower cells develops into the food which act as a taking the food material from the where there is often soils. Okay, that is a gametophyte. The upper cells which going develops into the capsule first divided into 6 to 8 cells of filament. Maybe that is often periclinial divisions from the amphitheecium and endotheciums. Endothecium forms columella and the amphitheecium is divided into jacket layer and inner archisporium, which develops into sporomother cells. These by meiosis develops into spores turns into a elaters are formed at the apical regions and where it is in outer layer, the capsule is the operculum, which ruptures and liberates the spores. Spores swelling in the suitable substratum develop young protonema, which later develops into the young gametophyte. So this is the sphagnum. Yeah. Next, now you see. So we are going to another one. Yes. So here there is often evolution of in gametophyte. Here we are completed now Hepaticopsida, Anthoceratopsida, and Bryophyta. These three we are completed. But now, how the development of gametophyte from which one that is in Marchamtia 2. Before the Marchamtia, some more plant is there. Hepaticopsida to Bryophyta. What type of development you are observing? What type of structures you are looking in entire the bryophytic plant? So already you see the first one where it is in evolution of gametophyte. Yes. So first now generally here two types of theories is there. That is in retrogressive evolution theory and second one that is of in condensation theory is there. Condensation theory is there. Progressive theory is there and regressive theory is there. Two types of theories is there when you are studying in Marchamtia evolution of Bryophyta evolution of gametophyte. So already we know very well the bryophytes are non-vesicular plants growing. It is only where the moist, shady places, where the aquatic zones. Okay. See here, they have two phases in their life cycles. One it is in gametophytic stage. Second one that is in sporophytic stage is this. One it is in gametophytic stage. Second one that is in sporophytic stage. Alternation of generations. Gametophyte plant body. Second one that is of sporophytic plant body. But these are all the gametophytic plant body are not looking the same. So here we are looking different type of the gametophytic plant bodies we are looking. So first now you see when we are looking in Marchamtia. Okay. So when you are looking in Marchamtia, so this is in only prostrate system is there. Prostrate systems. Thallus like structures is there. Okay. It is only thallus undifferentiated plant. Even the sporophyte also, so it comes to little bit outside of gametophyte stage, and in spite of it is totally prostrate system is there. Prostrate systems, there is no differentiated between. There is no differentiate here the root and stem and leaf. Prostrate systems. So when you are going to the second one, when you are going to the second one, when you are going to the second, that is of in Anthoceras thallus. So here we are looking the sporophytes are comes to outside in that area in spite of that the gametophyte here, no, which one that is in air pores is options is there. Assimilatory is also options is there. Loss of assimilatory filaments and air chambers. When you are studying in Anthoceras, Markamsha, here we are looking assimilatory zones is there and air chambers is there. So when you are coming here, this one, in this one here, loss of air chambers is this, okay, and assimilatory filaments is this. That's why the genera occurring in moist soil show the typical structures of complex forms with air chambers, pores, assimilatory filaments. The reduction of air chambers in some of the species, okay, where it is in the some of the species that is of in where it is in Geomatoria, air chambers are absent in the species growing underwater as very moist places. Okay, this is the first point here. And next one, simplification of the pores is some complex uh, forms of Markamsia. Markamsia, we are looking the pore-like structures is there. So these pore-like structures are also, okay, barrel-shaped is there, barrel-shaped is there. Barrel-shaped both on the thallus and disc of archegoniophore and antheridiophore also. The barrel-shaped on the discus but simple on the thallus, okay. So disc of the thallus, the rixia, there is no pores. You see, this is a structures we are looking in which one that is of in hepatic opsida. How the gradually decreasing of these are all the structures. So when you are coming in, here it is in the of in Anthoceras. So these yes. So after the Anthoceras, so when you are going to which one that is of in Bryophyta sphagnums. The sphagnum gametophyte is a hyphae developed in this. Hyphae developed. Hyphae developed means, so here we are looking, see now that one is rhizoids is there. Okay. So, but here also you are looking rhizoids, but not this much of development rhizoids you are looking in this one. So here this is the rhizoids is there. After the rhizoids, here we are looking one plant-like structure, this stem-like structure is there. This is the development. We call this often progressive theories, progressive. How the gametophyte is going to from simple to complex. Here this is in progressive. So here we are looking on plant-like structures is there. So these plant-like structures you are looking in above attached to one leaf structures also you are looking, leaf structures also you are looking in that area. And also tip of the branch is also looking this reproductive organs, one perigonial leaves also looking in that area. So these type of the development we call that is in progressive theory. How the taking food material from here, that much of development we are looking in that gametophytic stage, gametophyte. So this is a progressive theory. So some people are telling this of regressive theory. So what is regressive theories? Regressive theory means, see this gametophyte stage is there, but here no the proper food. What happened here? All coming closely together, okay, and coming from closely together, the formation like this. So that development you are looking in this area. So that is in regressive theory series. Okay, here to here we call that is of in progressive theory. From here to here you are looking that is of in regressive theories is this. So you see here, one, the bryophytes have the two phases in their life cycles. So this one, progressive theory here. That listens for that. One, it is in a gametophytic stage. Second, that is of in sporophytic stage. The main plant body that is in gametophyte, it is a thyroid. Main plant body, it is a gametophyte. Yes, gametophyte is a, it is a thyroid in nature, and liverworts are differentiated into stem and leaves in mosses. Evolution of the gametophyte from the simple forms like erexia, and next one that is in advanced gametophyte simple forms erexia, advanced forms which one that is of like mosses. The sex organs or anthrid and archegonia are developed on this gametophyte. Evolution of gametophyte from the simple thyroid forms evolved erect leafy forms or vice versa is explained by two theories. First theory, that is of in retrogressive evolution theory, supported by Werdstein's and Evans, Kashyap, all these things. According to this theory, the primitive gametophyte of bryophyte is a leafy shoot as a moss, slowly reduced to thyroid from retrogressive evolution of gametophyte. The second theory is a progressive evolution theory. It is was proposed by that is of Campbell. According to this theory, is a primitive bryophytes was thyroid. From it slowly evolved as leafy shoot from like moss by the progressive evolution of the gametophyte. So from here to here, progressive theory. From here to here, that is in regressive theories. So two types of theories is there in that one. So simple to complex, complex to simples. Hepaticopsida to, where that is of in Bryopsida. So Bryopsida to Hepaticopsida. Hepaticopsida. This here, this is of in two types of theories is there. Yeah. Similarly, next one. Okay, now gametophyte is completed. After the gametophyte, already we know very well, gametophytes are produced to the gametes. So these gametes are fused, formation the zygote. So here these zygotes are produced to the new plant that is in sporophytic stage. Okay. So there are two well-marked alternates of generation life cycles of bryophytes. One, it is in gametophytic stage. Second one, that is in sporophytic stage. So now we know very well what is gametophyte. The second one, now we are going to the sporophytic stage. You see, this is the gametophyte. Formation the zygote. Zygote is produced with a new sporophyte. But now you see in Marchamcia, Anthoceras, and Polytrichums, all these sporophytic stage are not looking the same. Sir, here also we are looking different type of the structures is this. Okay. So yes, so how the development of sporophyte from Hepaticopsida to where the Bryopsida. Yes, so first now you see when you are looking in Hepaticopsida, the node diagram is there. First, now you see here sporophytic stage. Here the sporophytic stage means already we know very well this is of in three stages is there. One it is in foot is there. Second that is of in seta is there, and third one that is of in capsule is there. But all the sporophytes are not looking the same. So when we are looking in Rexia in hepatic opsida, so there we are looking in only capsule-like structures. There is no foot and seta. Just a little bit stalk-like structures is there. A little bit foot-like structure it is not true. For example, this is the thallus. Sir, this thallus is attached to thallus. This is sporophyte. This sporophyte is attached to where the thallus. The sporophyte, there is no foot, there is no seta. So it is only round-like structures is this. Inside the sporophytic stage, there we are looking spores is this. Here, how these spores are comes to outside in this area. Sir, if these spores are not comes to outside in this area, next there is no generations. That's why when this cover is destroyed, the spores are comes to outside in this area. Similarly, when you're looking in Markhamsia, sir, Markhamsia, it is a foot-like structure is there, seta structure is there, and capsule is there. Capsule is there. Foot is attached to where the where the gametophyte. Seat, it is a stem-like structures, and here it is often capsule. Here it is in capsules. So these spores are comes to outside in the chamber. How means here some of the spores are modified as elaters. So these elaters are pressured. So some of the spores are comes to outside in this area, outside in the capsule chambers. Okay, and so next point. So Rexia 2, you see that much of development we are observing in that one. Rexia, there is no foot, there is no seta, and only here we are looking at the capsule-like structures in this. When this jacket layer destroy, spores are comes to outside in this area, that is of Rexia. But when you are coming in Markhamsia, so there you are looking some elaters-like structure. This is the development. Elater structures is this. Okay. Yes. So next, now you see. So when you are coming in, that is the last one. Second is of Anthoceras. So Anthoceras. We know already. In Anthoceras, here it is in a little bit developed. The two celled embryo is converted to 8-celled embryo and arranged in 2 tiers of 4 cells each. The lower tiers by further divisions forms massive foot-style structure is there, and upper it is divided to meristematic zones. So here this is the most important. So these meristematic zones again and going to divisions. So it is taking the food material from the, so it is prepared by the own food, That's why from here to the sporophytic stage is going to as a independent plant. Why means it is prepared the own food. That's why this stuff in second one that is in Anthoceras. And last one that is of Funaria, moss plants. Moss plant, it is an advanced group of entire the bryophytic plant. So here you see this sporophytic stage, high-fi advanced development. The foot is there and seta is there. The capsule is totally complex structure is there. Complex structures. Rexia, very simple structure is there. And Markhamsia, a little bit Okay, elater structures you are observing that one. Anthoceras, okay, their meristematic zone is there. So when you are looking in funaria, okay, so funaria, it is a apophysis is there. In between theca is there, theca. Okay, apophysis, it is a photosynthetic filaments containing the photosynthetic filaments, and in between that is in theca structure is contained the spore cells. Spore cells is there. Okay, and above the part operculum is there, operculums. So from food Sita, okay, so it is acting as a stem-like, it is taking the food material from the thallus. So it is giving to food materials where that is a capsules. So that capsule after the development, the spores are comes to outside in that area. Okay, so this is the development of an sporophyte from Markham's Rixia to where it is in, that is in Funaria or Polytech, whatever it is. So this is often progressive theories is there. Okay, and similarly same that one is reduction theories. The reduction theory means from here to here, that is in reduction theories. Okay, yes. So next now you see here once again inside the sections. The sporophyte is developed from the diploid zygote after the fertilizations. The sporophyte, okay, the sporophyte of Bryophyta is simple, soiled object and radial in construction without any appendages, as in Rixia, or it may be differentiated into foot and a seta and capsule, as in mosses. Okay, two role attached to the gametophyte. It only function production of dispersal of spores. Regarding these evolution of sporophyte, there is two main theories is there. One of theory of sterilization proposed by Bohr of fundamental principle involved in progressive sterilization and potentiality of fertile cells, sporogenous cells. The supporters of these theory were Campbell and Smith. The second theory, that is often reduction theories. In this, evolutions is in downward direction. The reduction is accompanied by simplification of structure of sporophyte. On the basis of this theory, the simplest sporophyte of Rexia is highly evolved. This is theory was supported by Kashyap. So this is a structure of in reduction theory in Bryophyta. Okay, so it's enough today. So tomorrow, so we'll go to the next one, that is in